So today we're going to keep talking about marine life, and probably for the last half of the period we'll start talking about what controls how much biological activity is going on in a particular part of the ocean. At first, at least, we're going to focus on open ocean processes, but eventually we'll start talking about other kinds of community as well. All right, at the end of the day on Monday, we started talking about primary production in the ocean, the role of organisms like phytoplankton or on land like plants that can actually harness energy from sunlight and use that energy and some inorganic chemicals in the environment to actually create stored biological energy in the form of things like sugars. Most commonly by photosynthesis. It's not the only way that organisms can convert inorganic materials into stored biological energy, but it's by far the most common, particularly in the oceans. And the basic idea for photosynthesis is that it largely makes use of one or several forms of chlorophyll, which is a pigment, so it absorbs light and thereby takes on a color. And in the particular case of chlorophyll, it's capable of absorbing light that's either red or blue, so either short wavelength or long wavelength light. And the light in between, that's sort of a wavelength on the order of 500, 550 nanometers. That's right in the middle of the visible spectrum, corresponding roughly to green light. Chlorophyll doesn't really absorb that light very effectively, and so when light passes through organisms or a group of organisms that have chlorophyll in their bodies, green is the light that's left over, and so they appear green to us by and large. This raft of sargassum weed in the Sargasso Sea in the North Atlantic Gyre is photosynthesizing because of the chlorophyll that turns it kind of greenish colored. So the basic idea of photosynthesis is it's a reaction that takes inorganic molecules, carbon dioxide and water, uses sunlight energy to convert them into things like sugar, which is a chemical that stores energy in a way that other organisms or the photosynthesizing organisms themselves can use, and produces oxygen as a byproduct. And rough ballpark number I want you to remember for this reaction is sort of the basic chemical reaction of photosynthesis, is that it goes to the extent of something like 100 grams of carbon per square meter of ocean per year. And if you add that up over the whole ocean, that's actually a lot of productivity. Respiration, which is what we do, is the opposite reaction. So that's the conversion of things like sugars back into inorganic compounds, releasing the energy that was stored in them by the photosynthesizers in the first place so that we can run around and do what we do. And so it's a disassembly process, and it uses up oxygen and releases the chemical energy. So its byproducts are essentially the same things that were used to make that stored energy in the first place, water and CO2 and some other things as well. And it's important to realize that even plants, even the organisms that do photosynthesis, the reason they're doing the photosynthesis is not to feed the things that eat them, but for their own purposes. They want to store the chemical energy so that they can reproduce, so that they can grow, and we kind of live off of the excess. And it's also important to realize that animals aren't the only things that do respiration. In addition to photosynthesizing organisms, many bacteria and archaea, which do not, in some cases, do photosynthesis, are also capable of doing respiration. Their metabolisms can be really diverse. So some of them actually respire in ways different from the ways we do. They may not actually use oxygen, for instance. They may use some other compound to burn biological molecules. So they may use iron, for instance, oxidized iron, and reduce the iron to a different form instead of using up oxygen. So the respiration reaction is crudely photosynthesis backwards, where instead of having to put energy in with sunlight, energy comes out when those biological molecules are broken apart in the presence of oxygen. Okay, so we can define a quantity, primary production, which is essentially that number I gave you a couple of slides ago. How much carbon is being converted from some inorganic form, carbon dioxide, into organic molecules like sugars? And we can define it per unit area, per unit time. Depending on how we define those units, it can be production or productivity. And the basic idea is that this process is essentially based on reactions driven by sunlight. Although in some cases there is production, like in deep sea hydrothermal vents, that occurs in the absence of sunlight. Sunlight really dominates the energy budget here. All right. So are there any questions about this basic idea of photosynthesis, fixing carbon from CO2 into some organic form, and then respiration kind of being the reverse of that process to get the energy back out again? All right. So this is like one of the basic things about biology in our ocean, how much of it is happening, how much material is being chemically processed by organisms in the ocean. So that's a basic measure of how intense is biological activity in various parts of the ocean. It's something we really want to know. And we want to know it not just for that reason, but for some perhaps more parochial reasons. In the process of respiration, CO2 is released, and we know that CO2 is a greenhouse gas. We'll get there in the last couple lectures for this class, but just for the moment, many of you probably already know this. So CO2, when it's released by respiration, can actually affect the climate and affect our own lives, even if the respiration is happening somewhere else on the planet. And photosynthesis is the reverse of that process, is one way that CO2 can be removed from the atmosphere, for instance. And of course, when we harvest seafood from the ocean, be it fish or shellfish or seaweed, ultimately all of that food energy is derived from photosynthesis. And so we're interested not just from the point of view of global climate change, but also from the point of view of understanding how much stuff might we be able to get out of the ocean without overwhelming its ability to continue functioning as a biological system. And this turns out to be quite a tricky process. It's a basic quantity, how much carbon is being converted from inorganic to organic. But it turns out to be kind of tricky in the ocean in particular. So we're interested in the same thing on land. In fact, we're really interested in the same kinds of processes on land. Anybody who runs a farm, the basic thing they want to know is, how many crops am I actually growing on my farm? And so it's very important for them to measure, how much photosynthesis is happening. And they may do it by measuring, what's the yield of tomatoes or corn or whatever is being grown. In the case of a forest, it's fairly straightforward also to, at least relatively speaking, straightforward to figure out how much photosynthesis is occurring in net, because plants like trees live a long time. So you could go to a field that's just been clear-cut, for instance, go to a forest, and try to measure how much mass of carbon is actually in the tree trunks and in the leaves and in the soil, and come back a year later and see how much different that amount of carbon is. How much bigger have the trees gotten? How much more carbon has been stored in the soil? You can sort of come back and weigh it later and determine the productivity by difference, at least crudely. Now, those measurements have some problems. It's really time consuming. You have to make some assumptions. It's not easy to pull a tree out of the ground and put it on a scale. So you have to figure out how to infer the mass of a tree, for instance, from its circumference or its height. And of course, these measurements are local. You go to a particular plot of land, a particular farmer's field, and you can measure the productivity there, but it's not so easy to get a global picture unless you make lots and lots of observations or find some way to distribute them so that you sample what's going on the planet as a whole. In the oceans, this gets even worse. And the basic problem is that, particularly in the open ocean, the organisms that do photosynthesis are more or less plankton, mostly. There are communities when you get into shallow water that aren't like that, coral reefs, for example. But in the open ocean, it's these little microorganisms that are doing most of the work. And those organisms don't actually live very long, and they don't stay fixed to a particular spot in the globe. They drift around wherever the water goes. And so you can't really go to a plot of water, scoop up a bucket, filter out all the organisms that might be in that water, weigh them, and then put them back in and come back a month later and pull out the same bucket of water. That same bucket of water no longer exists in one place. And the organisms that were originally in the bucket you pulled out have probably been eaten or reproduced several times by the time you come back. And so it's actually not trivial to figure out how quickly these organisms are photosynthesizing in the open ocean because they live such short, drifting lives. It's difficult to pin them down to do the accounting. There are some clever ways to actually get around this problem, one of which is kind of an indirect process. Okay, we can't just bulk determine very easily how much additional carbon has been added to a bucket of water because the same bucket won't be there in a week. But we might be able to infer it by, for instance, putting some inorganic carbon into the water that we can track. For instance, by putting inorganic carbon, CO2, or carbonate into the water that has a very rare isotope that doesn't really occur naturally very, very commonly, like carbon-14, which is radioactive. And so it's very rare. Something like 1 atom of carbon out of every trillion or so is carbon-14. It's really, really rare, but we can make it in nuclear reactors and stuff. And so we can add a tiny little bit of inorganic CO2 with extra carbon-14 in it and sort of dominate the carbon-14 budget for that entire system over some large volume of water, and actually monitor how quickly is that inorganic carbon-14 being incorporated into biological material, right? And that gives us a sense of, at least for that one rare kind of carbon, how quickly is it being cycled from inorganic to organic, and we could actually go the other way if we wanted to also. So this is a clever way of, instead of having to weigh everything in bulk, we can just monitor the transfer from inorganic to organic using a rare isotope of carbon. And this actually is one of the best ways to figure out how quickly photosynthesis is working, the turnover of carbon is working in systems like the ocean, where it's mostly being done by microorganisms that don't live very long, that drift around. However, it still has the same problem as weighing a forest or weighing a cropland. It's a local measurement fundamentally, right? You go to a particular spot, you sample some water, you do what you're going to do to it, and you figure out the productivity of that particular body of water at that particular time. It doesn't give you a global picture unless you do it a lot, and it doesn't give you a picture that's updated quickly with time, right? You have to go every time you want to make the measurement. You have to go and make the measurement. You don't automatically know what happened in December unless you happen to have made a sample in December. So it'd be very useful to have a method of determining productivity in the oceans where you didn't actually have to go out spot by spot and make these kinds of measurements. And of course, it would also be nice if you didn't have to use something like carbon-14 to make a measurement every time you were interested in doing it. It costs money. Carbon-14 requires nuclear reactions to be formed, and so it's kind of expensive to do that everywhere in the ocean all the time. So we want an indirect method. And so it turns out that the leading way of measuring productivity in the ocean globally and repeatedly as a function of time is actually using this property of the photosynthesizing chemicals themselves, that chlorophyll is a pigment. It has a characteristic color. It absorbs blue, it absorbs red, it leaves the green behind. So if you look down on a part of the Earth's surface where photosynthesis is happening, or at least where the apparatus to do photosynthesis is present, you're likely to see that as a green color. Turns out that water in the absence of chlorophyll is kind of bluish, right? And soils or rocks in the absence of chlorophyll tend to be kind of brownish or white. And therefore, when you see green, it's a reasonable inference that the green you're seeing is actually from chlorophyll, and you can actually look at the spectrum in more detail and see, do you see that peak of absorption at blue? Do you see that absorption at red? That is a fingerprint for chlorophyll. So it's possible to make indirect inferences about how much productivity is happening or what the capability for photosynthesis is just by looking at color and by doing some careful calibration work to make sure that the intensity of color actually corresponds to the rate at which photosynthesis is occurring. So the basic idea is really simple. You fly a satellite or a high-flying aircraft, you look down, you measure the spectrum of light coming back from the Earth's surface, and the greener it is, the more photosynthesis is happening, or at least the more capability for photosynthesis there is. Okay, so lots of green implies lots of chlorophyll, which you might expect would imply lots of productivity, lots of photosynthesis happening. And we have satellites that are in fact designed to do this, the Sea-Star satellite. And I've already showed you some pictures from that satellite. And essentially what you're seeing when you see a picture of productivity in the ocean over the whole planet is a processed image based on the color of the ocean. seen from space. Okay. So based on the fact that chlorophyll is what organisms use to do photosynthesis, and chlorophyll is green, we might hypothesize that the greener the ocean is, the more photosynthesis is happening. So what would we do? How do we test that hypothesis? Is it testable? If you wanted to check to make sure that green patches of ocean have lots of photosynthesis happening, what do you do? Yeah? Okay, so you could go to a spot in the ocean that looks green from space, and you could actually try to sample and see how many organisms are in that water. Is there a lot of photosynthesizing organisms present? Are there a lot of photosynthesizing organisms present whenever the ocean appears relatively green? And that's certainly something you could do. Anyone else? Well, you could do a cross-calibration. We've already talked about a way you can measure productivity in spots. For instance, by adding carbon-14 in an inorganic form to a particular part of the water and seeing how quickly it's converted to some organic form of carbon. And then if we had the satellite flying over the same part of the ocean, we could maybe determine, aha, here's what the Wade productivity is, the Wade rate of photosynthesis. Here's what the greenness is. Hopefully those two things correlate. And maybe we don't have these measurements everywhere in the ocean because we can't get to all the parts of the ocean. But at least in those places we can check, we can figure out, is the greenness correlating with the rate of photosynthesis measured by tracking carbon through the system? And this is what's been done. Is a cross-check. Is the greenness, the productivity inferred from a satellite, correlated with the amount of photosynthesis you actually measure on the ground by getting your hands into the water? And if you do that cross-check, this is what you see. In fact, this has been done, I think, more than once. This is just one particular figure. So what's being plotted here, this is a scattergram. And on the y-axis is the amount of photosynthesis that's inferred from the greenness of the ocean, essentially. There's some more stuff that goes into the calculation. You have to know something about what kind of organisms are present. If you're dealing with a photosynthesizing organism that lives fairly deep beneath the ocean surface, of course, the light, in order to get to the satellite, has to go through the ocean to get to the organism, interact with the chlorophyll to become green, and then get back out again. And if it's living deep, it might have trouble getting back out again. So all things equal, an organism living fairly deep in the water doing photosynthesis may not return as much green as an organism living right up near the surface. So you have to make some corrections for things like that. What's plotted on the x-axis is actually the Wade, indirectly or directly, amount of photosynthesis that's happening in the same patch of ocean. And there's a very good correlation here. 1-to-1 line is what this blue line is indicated. And that would be if there is a perfect agreement. If the amount of greenness and the modeled photosynthesis from the amount of that greenness was exactly the same as the amount of carbon fixation you measure, all of the points would fall right along that blue line. They don't all fall exactly there, but there's definitely a pretty strong correlation between the two. Okay, and we can actually calculate a correlation coefficient, which is pretty high. And so we can say, plus or minus 20% or something like that. The greenness and an inversion of that greenness of the ocean inferred from space actually tells us how much photosynthesis is happening in the water. And it's a much easier way to make the measurement because from a satellite you can see the entire planet, or at least a fair chunk of it at any one time. And a satellite's moving very fast, so it can cover a lot of the ocean. And it can cover a lot of the ocean over and over and over again. All right, so putting that together, we get a picture something like this. So this is an annual average primary productivity for the world's ocean inferred from the greenness of the ocean measured from space, from the Seastar satellite using CWIFS, which is one of these instruments that measures the color of the ocean. And you can see, first of all, that, as I said, something like 100 grams per square meter per year of carbon is a fairly typical number. A lot of the ocean is kind of that lightish blue color. Okay, and that's kind of the average. But it's not the same everywhere by any means. What parts of the ocean tend to have very high productivity? What? Yeah, coastlines, right? Almost everywhere there's a coastline, you see fairly hot colors indicating lots of carbon being fixed by photosynthesis. What parts of the ocean have low productivity? That's kind of true, yeah. So if you look at some of the colder areas, like down here maybe, right, tends to be bluer. Where else? Okay, open oceans in general are certainly less productive than coastal oceans, but is there a particular part of the open ocean? I'm sorry? Okay, areas of upwelling would be like along the equator, right? Along here, along west coasts. Lots of upwelling along here. Those tend to actually be really high productivity regions. That's right. So if you look inside the subtropical gyres, like the North Atlantic gyre, which goes right around like this, there's actually a desert in the middle of it, a region of very low productivity. And that's actually true of all the gyres. Inside the gyres, you tend to find very low productivity. So it's not just the open ocean, but it's in particular parts of the open ocean where you have surface water that's actually stable at the surface over long periods of time. Remember, in the gyres, they're high pressure systems, so surface waters are actually kind of being piled up in the center, and they're relatively warm areas of the ocean because they're at like 30, 35 degrees north and south of the equator. And so the water at the surface is fairly warm. It floats on top of the cold water that fills up all the deep ocean, and it's difficult to mix those two vertically because there's a pretty big difference in density between them. And so what happens in those areas where the surface water is stable for long periods of time at the surface, organisms do photosynthesis there. Some of them die and sink out of the surface ocean, and they take the nutrients with them. And over time, that surface water becomes depleted in nutrients and becomes a not very hospitable place to do photosynthesis. In areas where there's upwelling, in areas where you're near a continental margin, in an area particularly where like a river or agricultural runoff is coming into the ocean with fertilizer in it, lots of productivity. So what we're seeing here is actually, to first order, the distribution of nutrients in the ocean, which tracks how close you are to a continent and how long has that surface water been at the surface. The longer it's been at the surface, the less productive it becomes. Okay. Oh boy, this looks terrible. I'm sorry for that. Yeah, let's see if I can fix that really quick. Okay, maybe that's a little better. Okay, hopefully it's legible now. So we can actually make these kinds of measurements both on land and the ocean, divvy it up depending on what kind of habitat it is, crudely speaking. So these are pretty broad brush divisions, and what's plotted on the x-axis is essentially, again, this amount of productivity. The units here are actually a little bit different. It's not grams of carbon per square meter per year, but it's something that would be related to it by some constant of proportionality. So we're looking at relative differences here. And so what is an example of a high productivity environment? Well, things like forests, particularly rainforests, swamps, and it turns out estuaries, coastal parts of the ocean, tend to have very high productivity, lots of photosynthesis happening. But other areas of the ocean, and in particular the open ocean, tend to have fairly low levels of productivity relative to other ecosystems on the planet. Now, it turns out that the open ocean plays a significant role in the global budget of photosynthesis, even though it's not a terribly productive part of the ocean square meter by square meter. It makes up so much of the surface of the Earth that small productivity gets integrated over a very large area and ends up being quite important for the global budget. Continental shelves don't make up that much of the ocean, but they're highly productive by and large, and so they actually make up a significant amount of the productivity of the ocean, which is good for us because it means if we want to get fish or something like that, we don't actually have to travel out to the middle of the ocean to do so. At least we didn't, as long as we are taking things from the ocean at about the same rate it can provide them. Okay, so let's think about this. What might be causing these differences in the amount of carbon being photosynthesized in different parts of the ocean? Well, you need the driving energy for photosynthesis to happen, right? And that's really sunlight. That's by far the most abundant source of energy to do primary production in the ocean. And so you need sunlight. Well, okay, most of the planet gets sunlight some of the time, but if you're thinking about an area like in the Arctic Ocean that's usually covered by ice and that's dark 6 months of the year, that's going to place some restrictions on how much photosynthesis can happen there. If you're in an area that's really cloudy, that might also conceivably restrict the amount of productivity. And it certainly restricts productivity to the top of the ocean, the surface, 100, 200 meters into which sunlight can penetrate. You also, turns out, need nutrients. You need these trace constituents that allow organisms to build the proteins and other parts, the other biological apparatus to actually make photosynthesis work. Carbon dioxide and water are present everywhere in the ocean to some extent, so those basic nutrients are usually available, but you also need some iron, you need some calcium, you need some other things. And in particular, the case of iron and nitrogen and phosphorus, they tend to be fairly rare in seawater and limit the ability of organisms to do photosynthesis. So that turns out to be a very limiting factor on the productivity of the oceans. How many nutrients are there in the surface waters that are actually receiving sunlight? In particular, how much nitrogen and how much phosphorus? So the regions that are highest in productivity tend to be areas where those nutrients are available. So areas near continental margins, if you're on a west coast, or actually everywhere to some extent, there's a chance of having upwelling when the surface currents are moving offshore, dragging deeper water up to the surface that has more nutrients in it. And if you're near a continental margin, there's a chance of runoff from rivers or whenever there's a storm, or even from the wind blowing dust out to sea. Those all carry some amount of nutrients in them, and those nutrients may get added to the surface waters to enhance biological productivity. Away from continental margins, places like the equator, where surface water is diverging because of the equatorial divergence, because of Coriolis acting on the two different sides of the equator, pulling up deep water that has nutrients in it because it's been sitting in the dark for a while, having organic material broken down by bacteria and respiring organisms releasing their nutrients back into the water. When that water comes back up into the sunlight, the nutrients are available to do photosynthesis, and so lots of photosynthesis tends to happen. And in fact, anywhere there's vertical mixing in the ocean. So near Antarctica, where the surface water is quite cold, almost as dense as the deep water in lots of places, it's easy to replace one with the other. And so there tends to be a good flux of nutrients from the deep water up towards the surface where organisms can take advantage of it. Similarly, in the northern hemisphere, once you get up in the North Atlantic and the North Pacific, again, surface waters can be fairly cold. It's easy to exchange them with deep water or deeper water that has nutrients in it. The regions of lowest productivity are essentially those areas of the ocean that don't have any of those things happening. If you're pretty far away from the continents, if the sun's down because it's winter and you're near the poles, and particularly if you're in the middle of a subtropical gyre where surface water just kind of piles up and stays there at the surface for long periods of time, the nutrients get lost when organisms sink out of the sunlit part of the ocean, and so over time it becomes more and more difficult for photosynthesizing organisms to get the nutrients they need to grow. Okay. So we've talked about this productivity essentially as a chemical process without really thinking about the individual organisms. We've thought about it in terms of we have sunlight, we have water, we have CO2, we have nutrients, we add sunlight, and we make stored biological energy, and we can see this happening because the protein that does the process is green, right? But we haven't talked about what actually has the green in it. What are these organisms, and how do they actually function in the ocean? So we're going to spend a little while going into some more detail about some of the producing organisms in the ocean, the photosynthesizing organisms in particular. And we can sort of break this up both biologically and by habitat. And those two things actually kind of follow each other to some extent. So some of the primary producers in the ocean that we're most familiar with would be like seaweed or kelp. And those are eukaryotic algae, so they're eukaryotes. They're actually in the same domain of life as we are, and some of them are actually big organisms. That makes sense. And most of the seaweeds we're familiar with actually are benthic organisms, which we haven't gotten to that really in much detail, but you're going through it this week in your lab. A benthic organism is one that is either attached to or lives close to the seafloor. And in the case of seaweed, kelp are actually attached to the seafloor. So they're only going to really be able to do that if they're living in a part of the ocean where sunlight penetrates to the bottom. So there's a basic habitat requirement there. There are also organisms that are plankton, that drift around, that aren't attached to the bottom and may not even be associated with the bottom. They may live in a part of the ocean that's much deeper than sunlight can penetrate. And some of the groups of organisms that turn out to be important in that category would be diatoms, dinoflagellates, and coccolithophores, each of which we're going to talk about in some detail. There are some other organisms that are worth mentioning, even though I'm not going to be able to dwell on them very much. There are cyanobacteria, which I showed you some pictures of, those kind of greenish filaments from last lecture. Sometimes they're called blue-green algae, but they're actually a kind of bacteria. And they live in places like Shark Bay. They actually live in many places in the ocean and in fresh waters and on land. And I'm not going to talk about them further because there's not actually that much to say, at least that I can think of. But they actually are kind of important. And particularly they're important if you want to understand what was going on in the ocean 1 billion or 2 billion years ago, because they're probably some of the first photosynthesizers that came into being. And they're also pretty good at dealing with ocean waters that are not hospitable to other kinds of organisms. So they may occur in areas where other organisms can't. And then finally, it's worth pointing out that there are actually some areas of the ocean, like hydrothermal vents, where primary production is happening, where inorganic compounds are being turned into stored biological energy in the absence of sunlight. Okay, that's not the only energy source that can be used to drive a biological system. So there actually are organisms like chemosynthetic bacteria that can make use of things like sulfides in their environment or other kinds of dissolved metals to make their living. All right. Yes? Hmm? Yes. Anything that's called phytoplankton is basically an organism that does photosynthesis. So it requires sunlight. Yes, so pelagic here, actually we'll get there in a minute. But yeah, pelagic here is kind of the opposite of benthic in a sense. It's an organism that lives in the water column as opposed to at the bottom of the ocean. Like this guy. This would be an example of a benthic organism. That's an abalone. Although in this case, it's not actually in the ocean, it's in an aquarium, but we're not going to be picky. Okay, so let's look a little more closely at different habitats in the ocean, following up on the question. So the critical thing here from the point of view of understanding what kinds of organisms live where in the ocean and where this biological photosynthetic process is happening most vigorously is light, which only penetrates partway through the ocean, through the very surface of the ocean. And how light actually makes it into seawater is dependent on what's in the seawater in the first place. If we look at a fairly pure sample of water or seawater that doesn't have very many things living in it, then light can actually penetrate something like a couple hundred meters deep, but different colors of light actually penetrate to different depths. So you can see here, this is a spectrum of light And sort of how far down it's penetrating corresponds to how far that color of light would make it into relatively pure water or seawater that doesn't have lots of organisms present in it or anything else that would intercept the light. And so what you can see is that red light and far purple light, they actually get absorbed very close to the surface. They don't penetrate very far into the ocean. It's the kind of bluish light that makes it the deepest. And any of you who have actually been scuba diving or even snorkeling, you've actually probably experienced this. When you're looking down into the water, when you get below even a few meters depth, everything kind of takes on this bluish color. That's because blue light is the only light that's actually penetrating that far below the surface. In fact, if you want to take pictures under the water surface, any significant depth, you actually need special filters to correct for the fact that most of the light is bluish, and so other colors can get distorted for that reason. By contrast, when we go into the coastal ocean, there's actually other stuff happening, and so light actually by and large doesn't penetrate as far, and the light that does penetrate is not general blue, but kind of yellowish or greenish. And you might want to think about why that might happen. We'll get there. So that's the fundamental division that we're going to be interested in. Okay, there's going to be the part of the ocean that's getting sunlight, in particular enough sunlight to do photosynthesis with, and then there's going to be the part of the ocean that doesn't. And that turns out to couple with another feature of the habitat, which is, as was mentioned, benthic versus pelagic. So benthic means having to do with the seafloor. A benthic organism, like an abalone, like kelp, like a flounder, would be an organism that lives near or associated with the seafloor. And a pelagic organism would be like tuna or plankton. Those would be organisms that spend most of their time up in the water, surrounded by water, not at the very bottom. Okay? And so organisms that are benthic, they may rest on the bottom, they may swim along the bottom. Organisms that are pelagic, there's a fundamental division between the organisms that just kind of drift around and the organisms that actually swim, that actually determine their position in the water. So just to reinforce that idea, basic division here is between pelagic and benthic. Pelagic organisms being organisms that are either drifting or swimming freely in the water. So a whale shark would be a good example, although again, this is a picture from an aquarium, so it doesn't quite count. And a benthic organism like coral, for instance, which spends at least its mature life cycle attached to the bottom, would be a benthic organism. Okay. So if we look at, for instance, a profile across the typical continental margin, or this could easily be a volcanic island as well, it doesn't have to be a continent, and look at where is the bottom, in particular, where is the bottom relative to the location of that depth to which sunlight can penetrate. And there's a basic division that's going to happen. When you get very close to coastlines in very shallow water, sunlight actually might penetrate all the way to the bottom. And in those kinds of environments, it is possible for an organism like a kelp, for instance, to actually live attached to the bottom and grow while still getting enough sunlight to do photosynthesis. In most of the ocean, however, because the average depth is something like 4 kilometers, sunlight doesn't penetrate anywhere close to the bottom. And so any organism that wants to do photosynthesis can't really live attached to the bottom anymore. It has to be drifting or swimming up in the water column close to the surface. And so that division between the part of the ocean that's so shallow that sunlight penetrates to the bottom and most of the ocean where that's not true is the distinction between the neritic ocean and the oceanic realm of the ocean. In the neritic ocean, sunlight penetrates to the bottom. In the oceanic realm, sunlight does not penetrate to the bottom. Photosynthesis has to happen up in the water. So those are a couple terms that you should definitely remember. Now, the vast majority of the ocean falls into that oceanic category. Relatively little of the ocean is shallow enough that sunlight penetrates to the bottom. So we're talking in terms of area, the ocean being dominated by that part where any organism that wants to do photosynthesis has to live somehow freely drifting or swimming in the water column. Can't live attached to the bottom. All right. Is there any questions so far? Okay. That division between neritic and oceanic is essentially a division by penetration of sunlight. So shallower parts of the ocean, something like 100 meters in most places, but it can be somewhat deeper than that, is a part of the ocean where sunlight penetrates, and in particular towards the surface, there's enough sunlight to do photosynthesis to sustain an ecosystem based on that fixation of sunlight energy. And we call that part of the surface ocean where there's relatively large amounts of the sunlight the euphotic zone. There's enough sunlight to do photosynthesis. There's a region of the ocean, since sunlight is being absorbed gradually as you get deeper and deeper, where there's a little bit of light present, but not enough to really do photosynthesis, certainly not enough photosynthesis to outpace the rate of respiration of that organic matter as it's fixed. And so we would call that the dysphotic zone. There's a little bit of sunlight, but it's not really an active photosynthetic region of the ocean. And then finally, when you get below that, there's essentially no sunlight, no photosynthesis at all happening. The only thing that can happen is respiration or some chemical-based primary production. And in the open ocean, that's where that photic zone penetrates most deeply, something like 100 meters, maybe a stretch a couple hundred meters. But when we get into the coastal ocean, it's even shallower than that. So an area near the coastline, sunlight might only penetrate 50 meters. And in fact, when we used to be able to go out on the UCLA research vessel, we would go out into Marina del Rey, which is, of course, coastal ocean. It's very coastal. It's actually inside the coast, but filled up with ocean water. And we would regularly measure, how far down can you see a white-colored disk when you lower it below the ocean surface, which gives you a sense of, how far is sunlight penetrating into the water. And we would regularly find that in Marina del Rey, sunlight would penetrate, 4, 5, 6 meters, maybe 10 meters tops. So actually, in coastal oceans, sunlight doesn't penetrate very far relative to the open ocean. Why? Does anybody have an idea why that might be happening? Yeah. That's basically right. Yeah. So what's happening here is life, okay? When you're in the coastal ocean, there's typically a fair number of nutrients present for whatever reason, and so there's lots of organisms doing photosynthesis. There's lots of chlorophyll in particular floating around in the water, and then just bodies of organisms which also interact with light. There are other pigments besides chlorophylls. And you can actually kind of see that in the color. So what we're seeing here is that in more or less pure water, it's the blue light that penetrates most deeply. But remember, chlorophyll actually absorbs blue light. It absorbs blue light, it absorbs red light. And so if you go to a part of the ocean that has lots of chlorophyll in it, like a coastal ocean, the blue light is actually getting absorbed pretty quickly by organisms. And what's left over is the greenish light, which doesn't get absorbed very well by chlorophyll, but it also just doesn't penetrate very far through water, period. And so the net is the coastal ocean tends to be greener, and the light doesn't penetrate quite as far. All right, I'll skip that. So any questions about that, color of seawater? All right. So we'll get started talking about the plankton that are basically required to be the organisms responsible for photosynthesis in most of the ocean. Most of the ocean's too deep for sunlight to penetrate to the bottom. Any organism that does photosynthesis has to be drifting or swimming near the surface of the ocean, can't be attached to the seafloor. And for the most part, that means that the photosynthesizing organisms in the ocean are plankton. Plankton being defined as organisms that live in the ocean that drift. They may be able to swim, but they can't swim so strongly that they can fight against currents over large distances. But they may be able to control where they are in depth, for instance. Plankton quite literally means drifter, actually. So these organisms are organisms that kind of go where the currents take them. They can swim down out of the sunlight. They can swim up into the sunlight in some cases. They may be able to control their vertical position in the water by changing their buoyancy, by making gas bubbles inside their bodies, or storing oils, or making shells that are denser than seawater to sink. But in general, they can't swim against the tide, and they can't swim against currents. And so plankton is by definition a lifestyle definition. Any organism that drifts or doesn't swim very strongly is part of the planktonic community. And so jellyfish would be an example of a planktonic organism. A mature jellyfish, it can swim, but it's not really swimming against the currents. It's just swimming to get around and find food. And bacteria or diatoms or foraminifers, which actually can't swim at all, are also examples of plankton. So here's some plankton, again, from Marina del Rey. Here's a quarter for scale. And so you can see, actually, these are little copepods, about the size of rice grains, which is pretty small on a human scale, but actually turns out to be kind of big in the plankton world. And the important thing to remember is that for most of the ocean, because the water is too deep for sunlight to penetrate to the bottom, plankton form the base of the food chain. That's where the biological energy is first getting converted from inorganic sources in sunlight into something that the rest of the organisms in the system can use. And so everything else ultimately depends on these planktonic organisms. All right, so we'll come back on Friday. We'll start talking about particular types of plankton, in particular diatoms, dinoflagellates, and coccolithophores. And hopefully we'll get the grades up either tonight or more likely tomorrow.